we are going through a topic by name B-cell maturation, differentiation, activation. B-cell maturation, differentiation, activation. So myself is Professor Kripanidhi, heading the Department of Biotechnology at Vignan's Foundation for Science, Technology and Research, giving the series of talks on this. Hitherto we have seen this overview of the immune system, immunoglobulins and antigen-antibody interactions. In this unit, on the fourth unit, we are now looking into the T-cell and B-cell development. Okay, so here the B-cell maturation, differentiation, activation, we are saying activation. B-cell is getting activated by the T-cell. Okay, the T-cell gets activated by the macrophage. Once the B-cell gets activated, it produces plasma cell and the memory cell. The plasma cell gives what is called immunoglobulins. So those are the immunoglobulins. Now everyone is getting cured in this COVID, what is it called, 19 break and COVID infection. So as a result, the plasma cell gives immunoglobulins and the recovered patient, convalescent serum, convalescent plasma is being taken. That means he is having rich of immunoglobulins. That immunoglobulins are being given to the other patient who is suffering. So that's what here the B-cell plays an important role. In the name B has come from Bursa capricius of bird, which is present in the bird's cloaca. Okay, other than that also you can say bone marrow. So B-cell is a lymphocyte. So now to familiarize B-cells, to know the maturation of B-cells, to appoint the sites where the B-cells are being produced, originated, to appreciate the clonal selection theory. Here the clonal selection theory is an important aspect wherein the noble laureate Nels Jerny, he has explained this. Okay, here this topic involves two noble laureates contribution. One is Susumu Nigawa, who has done the, what is it called, the immunoglobulin diversity. Immunoglobulins are produced only by the B-cell in the animal system. No other cell produces immunoglobulins. So the development of B-cell involved in adaptive immunity and their embryonic and adult immunity sites will be known. So the B-cell and the T-cell are there in the adaptive immunity, and the clonal selection theory depicting the proliferation of activated T lymphocytes to maintain adaptive immunity will be known. So adaptive immunity, these are the two cells, only the T-cell, B-cell. The details will be known here. Okay, in adaptive immunity, what is that? Our system should get adapted to one exposure. If we get adapted to this exposure, next time when you get exposed, okay, we will be having the memory. That's what experiential learning. As a result, these memory molecules called immunoglobulins, memory cells produced by the memory cells, will be useful to fight for the succeeding repeat infections. Blood cells are produced in the yolk sac and liver. Yolk sac is there in the embryonic development. And again, blood cells to produce, first to produce the yolk sac, later transferred to the liver. From the liver, after postnatal, it gets transferred to the bone marrow. Okay. So the stem cells gets differentiated into myeloid lymphoid series. If the B cell comes under the lymphoid series. B cell, T cell, NK cell. These three are coming under the lymphoid series. Okay. So that's what. The yolk sac in the embryonic. That's what embryo. From there it goes to the liver. From the liver to the thymus. Okay. And again here from the liver to the bone marrow. Those who go to the thymus, they become the T cell. Those who go to the bone marrow, they are going to the B cells. Okay. So this again from the bone marrow, from the thymus, the blood goes to the lymph nodes. Okay. So this is the lymph node where the blood traffic is slowed down, where a pathogen can be seen and encountered. So that what B cells go to this region and the T cells occupy here. So the lymph node is the one where encountering of the pathogen takes place. See how the memory B cells are getting developed. So now several of the patients who have been recovered from the COVID-19 infection, they are all having the memory. Second time they are not supposed to get the COVID-19 infection.
Today I would like to introduce to you about memory B cells and its development. As you might have known, human body have different immune cells that help us to fight infection. These cells belong to the adaptive immunity. They can accurately target the antigens and eliminate them from the body. Memory B cells are able to remember the pathogens after the first encounter and ready to fight them off faster and more efficiently in the next encounter. With such a high specialization, memory B cells undergoes an extremely complicated development, as illustrated in this diagram. However, this process can be generalized in 5 stages. Stage 1. It begins with the migration of the multipotent progenitor cells from the liver into the bone marrow. The progenitor cells continue to differentiate into lymphoid precursors and lymphoid progenitors that is responsible for the B cell lineage. Stage 2. Lymphoid progenitor receives signals from stromal cells resided in the bone marrow to induce the development of the broad B cells. These signals include PCAM that binds to BLA4, SCF that binds to c-KIT, and IL-7 that binds to IL-7 receptor. The co-receptors Ig-alpha and Ig-beta are also observed on the cells at this stage. Stage 3. The broad B cells develop into immature B cells with the expression of IgM on the surface of the cell. IgM and the co-receptor Ig-alpha and Ig-beta together are called B cells receptors. Stage 4. The immature B cells then leaves the bone marrow by expressing S1P receptor, which attracts S1P protein in the blood. It migrates to the germinal center in the secondary lymphoid organs, such as lymph nodes and spleen. Within the germinal center, B cells undergo rounds of division, affinity maturation, and somatic haplorotation. The class switch recombination from IgM to IgG, IgA, or IgE can also occur. Stage 5. The activation of B cells can be T-cell independent and T-cell dependent. Notice that T-cell independent activation does not result in the production of memory B cells. B-cell activation is initiated when the IgM and MHC2 of B cells binds to antigens. Then CD40 ligand on T helper cells binds to the protein CD40 on B cells. Other cytokines also help to activate B cells. The activated B cells then proliferate and differentiate into plasma cells and memory B cells. The memory B cells have a higher expression of membrane-bound antibodies and secretes mainly IgG in the secondary response. Memory B cells also have other markers such as CD27, which help to recall previous responses. And that's the overview for the development of memory B cells.
Hello. Just now I have gone through a wonderful animated video on B cell development, how the memory B cell forms, wherein there is precursor B cell, pre-B cell, pro-B cell, and immature B cell, activated B cell. In between there are several cytokines are coming. Ultimately, there is IgM to begin with, later IgD to begin with, later it is joined, and here CD40 ligand. So please remember, CD40 ligand prompts the mature B cell of this B cell to have class switch. There is somatic hypermutation takes place. There is, as a result, VJ are getting altered, one of the alteration comes. That's what called somatic hypermutations. That's what Snoop Toligava's contribution in this phenomenon. We will see those details now. So now this is B cell maturation and differentiation in bone marrow. Because I have been telling you from the yolk sac, the B cells come to the liver. From the liver they come to the bone marrow. Okay, the bone marrow they cannot encounter the pathogen. Bone marrow is a highly specialized place inside the femur and humerus, and there this pathogens entry into that is prevented because of the blood-brain barrier and all those things, something like that. Okay, so here from the bone marrow they have to go to the lymph gland. Before the bone marrow, before they go to the lymph gland, what happens in the bone marrow? You see now. So here in the bone marrow, this is having mu germline. Mu. That is IgM will be there. Along with that, there is kappa and lambda. That is immunoglobulin light chain, either it may be kappa or it may be lambda chain. So later, the early pro B cell. Okay. Later, late pro B cell. In the late pro B cell, when you come, there is a diversity gene comes, and joining gene comes. There is a diversity gene, joining gene, variable genes, and constant genes. One after the other, they keep undergoing somatic hypermutation. Right? The diversity joining will join here. Later, B, variable diversity joining gene comes in the large B cell. Later, small B cell, there will be mu, and again, this immunoglobulin is formed here. So ultimately, in the immature B cell, IgM is expressed. IgM is expressed on the B cell. Okay. Now let's see. What happens in the secondary lymphoid organs? Secondary lymphoid organs means they are the lymph glands. In the lymph glands, to begin with, it is having IgM. It's having IgM. Later, IgD is being added. That's what now immature B cell. Okay. Later, mature 9 B cell. Mature 9 B cell where IgD, IgM is expressed. So IgD expressed high, IgM expressed low. Now let's see activated, antigen activated B lymphocyte. Now that is the stage. It is going to meet T helper cell. T helper cell is going to bring its antigen along with the MSC molecule. This is the stage where it is getting activated B cell. Once the B cell gets activated, then B cell gives IgG, immunoglobulin. So these are the immunoglobulins that COVID patients are having. Okay, so once the IgG is prepared, and there will be a memory cell, there will be the memory cell, and then this memory cell is ready for future infection. Okay, so like that the B cell development goes. Now the B cell display surface IgM. That's what we are seeing, that IgD is being added. So B cells constitute 30% of the total lymphocyte population. Okay, so their lifespan is short. That's what in hybridoma technology they are mixing with what is it called myeloma cells. So the B cells undergo the clonal selection theory. The clonal selection theory was proposed by one of the Nobel laureate, Niels Zernig. Okay, so that's what they are going to explain now. So Niels Zernig, Burnett, and Talmage, they proposed this lymphocyte arise from the clots. That is, the plasma cell and the memory cell are not coming from all the B cells. We have got thousands of the B cells in our system. All thousands of the B cells are not responding to the COVID antigen. Only one or two. Which one is responding? Such type of the B cell proliferates. That's what called clonal selection. Okay. So that's what the clonal selection theory that we explained. So this is what the lymph node, where the B cells undergo the development. B cells undergo the development in this region. Okay. Now, let us see the clonal selection theory. That is the antigen. Let us say COVID antigen. So the COVID antigen binds to one of the B cell. Let's say B cell 1, B cell 2, B cell N. Okay. So once it binds, this number 2 cell alone undergoes the proliferation, not number 1 and number N. Okay. So this proliferation continues. And they produce these immunoglobulins. So these are the immunoglobulins which are rich in the what is it called, the infected person. Okay, even after the recovery from the infection, he will be having these things in the plasma that is being used for the treatment. That's called plasma therapy. Let's see the summary. Red cell precursors originate in yolk sac and liver and transform the bone marrow during the postnatal life. B cells display IgM followed by IgD. Okay, so they mature in the bone marrow and circulate in the blood and ultimately settle in lymph nodes. Activation of B cell undergoes the clonal selection. Once the B cell is activated, clonal selection takes place. As a result, the B cell which is activated becomes number of B cells, clone of B cells. Okay. IL-4 and IL-5 promote lymphocyte proliferation. IL-4 and IL-5 are released by the B cell. They promote this lymphocyte proliferation. So let us have this study questions here. Elaborate the differentiation of B cells development. Where from the name B came for the B cells. Okay, it must have been labeled as ACD. Explains cell cross switch in B cell development, indicate the immunoglobulins gradually added to developing B cell. IGs, then IgM, IgB, IgA. Elaborate on B cell differentiation. What is the attribute of the B cell? What is the attribute of the B cell? What is the attribute of the T cell? Like that. Okay, get to know these answers. Let us sincerely acknowledge online sources and public domains for the use of the content to develop teaching material. So with this, the B cell development, activation, maturation, clonal development, and again how the memory B cell is formed, all those details we covered. Please go through. Thank you very much.